The last session we have discussed about passive transport on an MCQ based format. Today I'll be discussing about active transport across cell membrane. Active transport means one, it is energy dependent. It is an energy dependent and the energy is obtained from ATP. The transports can be primary active or secondary active transport. Primary means the energy is utilized at the site of the pumping, and secondary means energy is required for the purpose, but it is not at the site of the action of the. So the question is, which of the following is an example of primary active transport? So primary active transport means energy is utilized at the site of transport. Let us look into the little more detail regarding the sodium pump. So this is the carrier protein which is acting as the sodium pump. It has got an alpha, beta and gamma domains. The alpha subunits binds with sodium, potassium and ATP. Inside of the cell there is a site for ATP. Outside there is a domain for obein. Obein is a cardiac glycoside which can inhibit the sodium-potassium pump. So once the sodium and potassium is attached, the ATPase activity for the pump occurs or the carrier protein occurs, and ATP is split into ADP and phosphate, and this energy is used for pumping sodium outside. Even though extracellularly sodium is high, is going against the concentration gradient. So, and ATP is used at the site, so we can say this is a primary active transport. Hope this is clear. Sodium pump is essential for the maintenance of the integrity of any cell. It is present in all the cells. Now we go to the next question. Which of the following type of transport is found in the small intestine facilitating glucose absorption? That is, when you consume a carbohydrate meal, it reaches the small intestine from where the glucose gets absorbed. So let us take a cell from here. This is the luminal site and this is the cytoplasm, and we have got sodium potassium ATPase. So which pumps potassium outside, so and the potassium coming inside and sodium is pumped outside, so that the sodium concentration inside the cell is comparatively less. There is a concentration gradient for the sodium inside the cell maintained by the sodium potassium pump. So in the luminal site we have got a carrier protein which can attach sodium and glucose. It gets activated only when glucose and sodium gets attached. So what happens? When sodium and calcium gets attached to this protein or carrier or channel, sodium will move inside on concentration gradient because inside sodium is less. And how this gradient is maintained? It is because of the sodium potassium pump. So energy is required, but the energy is on an indirect way. Along with the sodium, glucose also enters. Glucose enters against a concentration gradient, because when the glucose is getting absorbed, the concentration of glucose inside the cell becomes higher than that in the lumen. So it is again a active transport, but it requires energy, but the energy is occurring because of the sodium pump that produces a decrease in the concentration or lower concentration of sodium inside the cell. This sort of transport is called a secondary active transport. So the correct answer is sodium glucose transporter. We have already said that the sodium pump is an active primary pump, and glucose transporter, but it is part of a facilitated diffusion. No energy is required for that, which we have discussed previously. So this is a little more better picture. You can see here that because of the sodium-potassium pump, inside the cell concentration of sodium is less. And this is the SGLT, sodium glucose. Sodium goes inside because of the concentration of sodium inside is less. Along with that, glucose enters. So you can see that both these are entering in the same direction. So it is a co-transport or symport. So materials are passing in the same direction. It is called the symporter. This SGLT2 is present in the renal tubules also for the absorption. Again the same mechanism. The concentration gradient is maintained by the sodium-potassium pump because of the ATPase activity, and the transportation, reabsorption of glucose from the luminal side, that is from the proximal convoluted tubule. Sodium enters along because of the concentration gradient, and the glucose also moves along with the sodium. That is important. And in the management of diabetes, we can use a blocker of this, that is the SGLT2, so that the glucose will be passing through the urine and can control or utilize for controlling. So SGLT2 is used for management of diabetes, blocker of SGLT2. Now we go into the third question. In secondary active transport, which of the following describe co-transport of two substances in the same direction along the cell membrane? We have already answered this. This is symport. That is, two substances are passing. So here you can see a symport. So we have said about the sodium glucose is a symport. If one is going inside and other is going outside, it is called antiport. Only one material is going, it is called uniport. 3 times you should remember. Hope it is clear to you. So we look at an example of a antiport. So this occurs in kidney. In tubules it can happen. There is a sodium hydrogen exchanger. The lumen sodium is more. The concentration gradient sodium get inside, and the sodium gradient is produced because of the sodium-potassium pump. So it is an indirect active transport. And along the hydrogen ions moves out. So this is an example of an antiport. So here sodium is moving because of the concentration gradient, that is sodium is higher outside, lesser inside, and this concentration is gradient is maintained because of the ATP. ATP is activity, that is sodium potassium pump. That is why it is called an indirect active transport and is an example of an antiport. Now we go to the next question. A 3-year-old with cystic fibrosis presents with thick sticky mucus in the airway. What is the defect in the cellular transport most likely responsible for this symptom? Cystic fibrosis. The options are decreased sodium pump, impaired SGLT2, dysfunctional chloride conductance. The correct answer is dysfunctional chloride conductance. We will study about cystic fibrosis. This is a disease of the chloride transport along with water and some ions. So this is the cystic fibrosis transport protein, CFTR protein. This acts as a channel for the chloride ions. When there is a mutation in the CFTR protein, this will lead on to defective chloride conductance that can lead on to features of cystic fibrosis, about which we will study later. In the next session, we will discuss about vesicular transport.